My name is Iñigo Olalde. I am a Ramon y Cajal researcher at the Department of Zoology and Animal Cell Biology of the University of the Basque Country. And today I'm going to be talking about one of the most recent applications of archaeogenetics, which is recovering the genome of all the individuals from one archaeological site and understand all the familiar relationship and to know more about how past societies were organized. As you can see, it's family affairs 6,000 years ago. I'm going to introduce myself. I'm going to explain you a little bit my scientific career because, when I give this kind of talks, they tell me that it's quite useful for you to see like a typical scientific career. Maybe some of you are considering in a couple of years entering into a biology degree or science degree, are thinking about becoming a scientist. So this could be like a typical trajectory. So I'm from Vitoria. I studied in Colegio Marianistas. I don't know, maybe some of you know it. And when I was about your age, I started to like more things like biology and chemistry, physics as well. So I had some doubts about whether going to university for biology, for chemistry. At the end, I decided to study biology and biochemistry at the University of Navarra. I was there between 2005 and 2011. And the last year of my degree, I went for an Erasmus stay in the Netherlands. And there was a lot of party, like in every Erasmus every Erasmus student, but I also had my first experience working in the lab, working in genetics. I spent basically like 4 months locked up in the lab doing PCRs of stick insects, so Insectos palos. And I really liked the experience. I really like genetics. Maybe not. I was not considering doing a career in like stick insect genetics, but probably like in human genetics. So at the end of the grado, of the licenciatura, I decided to do a master's in advanced genetics in Barcelona. And of course, this was 1 year, and at the end, you start to think again, what are you going to do next? So I started thinking about it, and the next step is usually a PhD, so a doctorado in Spanish, and you have to start thinking about where I'm going to do it, in which topic. And by that time, there was the publication of the Neanderthal Genome Project. So this was around in 2010, 2011. There was this publication where they described the first time that they were able to sequence a complete genome of a Neanderthal. And it made it all the news. It was widespread in all the media, and it blew my mind because now we have many genomes from Neanderthals, but at that time it was the first one. I said, okay, this is how crazy this is that we can have the genome of a human species that got extinct like 40,000 years ago. So I don't know if any of you know this guy. Does any of you know who this guy is? So this is this Professor Svante Pavo is the director of the Neanderthal Genome Project, and last year he got the Nobel Prize winner. So at that time I decided to write to him to ask him if I could do a PhD, a doctorate in his group. And I don't know, what do you guys think? He said yes or no? Thank you for your confidence, but he said no, unfortunately. If he had said yes, I would be now, like my mentor would be a Nobel Prize. That would have been nice. But anyway, he said no, and I had to look for a different option. And in the Neanderthal Genome Project, there was a scientific group that was participating in this project, and it was in Barcelona. It was Professor Carla Lalueza Fox. So I also wrote to him, and in this case, he said yes. So I stay in Barcelona for a PhD, which usually is between 3, 4, or 5 years. And the lab is located in this building, so as you can see, it's as good as it can get for a place to work. It's like right in the Barceloneta beach. So it was a really, really nice spot, and I had a great time there. I dedicated my PhD to recover the genome of ancient humans, in this case, not Neanderthals, modern humans like us, but very ancient, like 6,000, 7,000, 8,000 years ago. And by the end of this period, we started collaborating with a group at Harvard University, which is one of the best groups doing ancient DNA, and is this one over here. And so I moved there for what is called the postdoctoral period. So after you finish your PhD, you have to do like the postdoc is called, and there you are a scientist already. So you are not a student anymore, let's say. And here I also had a really great time. I had the opportunity to work with amazing scientists, and just to give you an example, there is this guy over here, Nick Patterson, is a very peculiar character. He worked he's from the UK, and he worked in the UK Secret Services as a cryptographist. Then he moved to the US. He also worked, I think, at the CIA, working on cryptography. Then he moved to Wall Street. He worked for a hedge fund, for a fondo de inversión, and he applied statistical techniques to predict the behavior of the stocks, and he became a multimillionaire. And then when he was very, very rich, he decided to move to what he really wanted to do, what was genetics. And he's the guy who has developed a lot of statistical techniques that we now apply in my field. So it was great to work with him and with others as well. And of course, I was there for 3 years, and it was really nice, but as always, you start feeling morriña a little bit. So I wanted to come back to Spain, and then I did it. I went back to the place where I did my PhD, to Barcelona, for a year and a half, and then the COVID pandemics got me there. And then finally, I was able to do the whole trip and come back to Vitoria, where I'm from. And since 2021, I'm Ramon y Cajal researcher and also Ikerbasque fellow at the University of Basque Country in the Araba campus. So it's been like, closing the circle after 15 years traveling and working in different places. At the end, I was able to come back to my hometown. And the reason I say this is because this is like a typical career for a scientist. I have many friends working in science, and almost all of them spend some time in a different country, and almost all of them at the end want to come back. Some of them come back in the end like me, and others stay. But anyway, if you guys have any questions about a scientific career, you can ask me at the end of the talk, or you can drop me an email. And enough talking about me. Now we are going to go into the talk. And my field of research is called archaeogenetics, or paleogenetics as well. And it's a very peculiar field of research because it's right kind of in the middle between the biological science and the social sciences. So we use techniques that come from biology, from genetics, but to answer questions that come from social sciences and humanities. So we basically collaborate and also work in the same group with, myself, I am a biologist, but together with archaeologists, with historians, and also, as we will see, now we are working with a huge amount of data. So we also work with mathematicians, informaticians, physicists, and also when I was in Boston, for instance, in my group, there was a guy who was an engineer. So as you see, like, it's quite fun because you are able to interact with people with very different backgrounds as you. And what kind of questions do we ask? So one of them, and the one that we are going to be talking today, is how societies in the past were socially organized. And we can think about, going into a time machine 6,000 years ago and look at, for instance, at a birthday party and see who was sitting in the table. So it was maybe the guy who was celebrating the birthday party, but it was it his maternal family with him, his paternal family? Were there all the relationship monogamous, or there was polygamous relationship? Was that person living at his wife's place, or did his wife move with him? So all these different questions about social organizations are the ones that we ask in this particular topic. And And we definitely know some things about how societies were organized in the past because we have the historical records. We can think about 2,000 years ago, during Roman times, people used to write about the people that they encountered. And for instance, we have here one example, the famous Julius Caesar. He wrote a book which is called the Bello Gallico, and there he described all the conquest of La Galia, which is present-day France, and also a little bit about Great Britain. And he said, 10 y 12 hombres comparten esposas, en especial hermanos con hermanos y padres con hijos. Los que nacen de ellas son reputados hijos de los que primero desposaron las doncellas. So Julius Caesar is telling us here that these people, the Britons, were organized in a very different way as compared to today. In the Western world today, it's mostly nuclear families, a man and a woman with children. That's the most common type of organization, but these people were polygamous, and also they didn't care much about who was the biological father. So here he's saying that your father will be the first guy who had sexual relationship with your mother, but that didn't care whether it was the actual biological father or not. So they didn't care much about biological kinship in this place. And this is another example that Tacitus, which is a Roman historian, also tells us, talking about the tribes in present-day Germany. He says, a los hijos de la hermana se hace la misma honra en casa del tío que en la de sus padres. Algunos piensan que este parentesco es el más estrecho e inviolable, y cuando han de recibir rehenes les piden más que a otros, porque les parece que estos les serán más firmes prendas como más queridos, así en la familia del padre como en la del tío. So what he's telling us that for these tribes, the maternal uncle was a very, very important figure. And he's saying that, when sometimes they will be doing peace treaties. So there was a war, and at the end they sit down and then negotiated a peace. And, in order to make sure that the other part will fulfill everything that they have negotiated, I will give you, for instance, I was making a pact with you, I will give you my son, and you will give me your son. So if you don't fulfill the pact, then I could do anything that I want with your son. So he's saying that in this case, I will not ask for your son, but for the son of your sister, because that would be equally important for you and for the other family. So this is, again, a little bit different as what is today. And this is the last one that I'm going to talk about, and this is, again, pretty similar to what happens today. Today, when Pedro Sánchez goes to the US and meets with Joe Biden, then they have some negotiation about politics, but then during the night they have a dinner, a party with not only between them, but also with their wives and with other members of the family. So it was the same in Roman times. Dion Casio here is describing a meeting between the Roman emperor, which was Septimius Severus, and Argentocoxo, which was the leader of the Caledonian, which is a tribe in Scotland. And he's saying that the wife of this guy, of Argentocoxo, told Julia Augusta, which was the wife of the Roman emperor, she said, nosotras cumplimos con las exigencias de la naturaleza de una forma mucho mejor que vosotras, las mujeres romanas, pues nos acostamos abiertamente con los mejores hombres, mientras que vosotras os dais secretamente al adulterio con los más viles. So this would be one of the first tasks recorded in history, and he's describing that the women among the Caledonians were much more liberal in the way they approach sexuality. So they could have sex with different men, and that would be approved by society, whereas the Romans were much more conservative. So I'm pretty sure that women had sex with other men that they were not the husband, but it was done in secret, because that was not a good thing for this society. And we could have way more examples about this type of information that the historians tell us, but being very optimistic, we could go back maybe until 2000 BC, so 4,000 years ago, because before that, people didn't write. So we basically, for 99% of human history, if we assume that modern humans appear in Africa around 150,000 years ago, for 99% of human history, we basically know nothing or very, very little about how human societies were organized, about this type of information. But now we do have a tool to understand this, and this is thanks to the what we call the archaeogenetic revolution. And what we do is the following. So we have the archaeologists excavate archaeological sites, and sometimes they recover human cemeteries, and they recover several skeletons. So what we can do is to take a piece of bone or a piece of tooth for each of these individuals. We can extract the DNA, recover all the DNA in these bones. We can sequence the whole genome of these individuals with the new sequencing techniques. And then we can do bioinformatic analysis and establish what was the relationship between each of these individuals. So was maybe the children was the son of this woman and this one was the father. So we can put all of them in a family tree and then study how we're all socially organized. And this archaeogenetic revolution is a technical revolution. It's the same as the microscope revolution. So what happened with the microscope? So somebody invented the microscope, and suddenly there was a completely new world that we can study. Before that, it's not possible to access. Also, with the telescope, the same thing. When the telescope was available, suddenly the whole universe could be studied, and before it was impossible. And with the archaeogenetic revolution, is pretty much the same. It's a technological revolution. It's a new way of sequencing the DNA. With the previous techniques, it was not possible to recover the whole genome of ancient individuals. But with these second-generation sequencing techniques, now we can access this huge amount of information that is hidden in the bones. So we can sequence all this DNA and establish what was the relationship between all these individuals that without this, we can only guess. We can only guess whether these are related or not. But now we can test it with these technologies. And just for you to know, how transformative this revolution has been, with the previous generation of techniques, with the so-called first-generation sequencing techniques that are based in the famous PCR. We all know now what a PCR is. So these techniques are based on the PCR. There was the first ever human genome sequence was with these techniques, and it was the Human Genome Project that finished in 2004, and it took 14 years. And I don't know if any of you guys can guess what was the cost of this, of just sequencing the first human genome ever. You can guess, just give me a number, for instance.
20 million.
20 million, much more. Come on, guys, just guess. 50? No, way more, way more. More, por allí.
2,000 million.
But more. So it's around $3,000 million that cost just one human genome, and it took 14 years. And now, with the second generation techniques, we can sequence the human genome in just maybe one day, and it costs $500. So it's from $3,000 million, or what the Americans will say $3 billion, up to $500. So before, it was technically possible to sequence a human genome, but in practice, nobody could because, like research labs in the universities, they never have $3,000 million to spend. So it was not possible to sequence a human genome, and even less to sequence an ancient human genome. But now with these new techniques, it's in a price range that we can afford. And now we, for the first time, are able to sequence whole genomes from present-day individuals, from modern individuals, but also from ancient individuals. And now I'm going to show you very quickly how an archaeogenetic project looks like the ones that we do in my lab. First, it always starts from archaeology. So our archaeologist colleagues that excavate an archaeological sites with a lot of effort, and sometimes in very bad conditions. Can you imagine being like this in the summer, 30 degrees, in this position for hours? So it takes a lot of work to excavate, and sometimes they found skeletons like this time. So we established collaborations with the archaeologists, and then to analyze these human skeletons. And we take the samples to the lab, and as you can see, we do it in sterile conditions because we don't want to contaminate the samples with our DNA, because our skin is full of dead cells, full of DNA, so it's really, really easy to contaminate the samples. And what we do first is just we take a very, very small sample. For instance, if we have a tooth, we will do a small hole and get just 30, 40 milligrams of bone. This is nothing else. And I have showed you guys just a real tooth that we have worked with. This is a tooth from a woman who lived around 5,000 years ago and who was buried in a cave in Guipúzcoa. And you can see, you can pass it. Just what we did is this very small hole in one of the roots, and that's all we need. We don't need to destroy the whole tooth as with the previous techniques. You have to destroy the whole tooth. Now we only need 30, 40 milligrams, so very, very little. And then what we do is we separate the DNA from the other materials that are in the bone, and we do this with some chemical treatments. And then we go and sequence the DNA with these Illumina sequencers, with second-generation sequencing. And what do we get from the sequencer? So it's pretty simple. We get a text file, like the ones you can see in your computer, with thousands, millions and millions of DNA sequence. Each of these DNA sequences comes from one DNA molecule that was in the tooth. And this would be a file with, thousands of millions of rows. So what's the problem here? It's impossible to open with your desktop computer. It will crash immediately. So we are talking here about massive amount of data. Each individual, you will get terabytes of data. So we need to work with supercomputers. So you see, you will be in your computer, but actually you connect with the supercomputer and you send all the data and all the processes. So it's necessary to know how to program, to how to work in the command line and so on. So it's genetics is not anymore just that you will work in the lab. It's actually most of the time. In my case, for instance, I spend 90% of my time in front of the computer analyzing data. It's not anymore a field that you will be all the time in the lab working there. It's just you do some part here, but most of the time it's just bioinformatic analysis. So if I can give you one advice, guys, is if you are considering working in science, if it's in biology and genetics for sure, but just learn how to program. You don't have to be a hacker, but some basic programming skills will be super helpful, even if you don't go into science, into other fields now, but definitely if you want to go into science. And now we are going to talk about one archaeogenetic project that I was involved. I think it's a very beautiful one, and show us what you can actually learn. This archaeological site is called Hasselton North. It's a huge megalithic structure, a funerary structure, similar to the ones that we have here in the Basque Country, the dolmenes, but it's much, much larger. And it's located in the south of England. It's dated about between 5,000 and 6,000 years ago. And here you have a picture. Here they were just starting to excavate. They had just removed the top soil layer, and as you can see, it's huge. This will be the people who are excavating, so you can see the difference in size. And you probably also notice that this is in a crop field. It's un campo de cultivo. So every year when they were plugging the field, cuando estaban pasando el arado, they will damage the structure. So they decided to do a rescue excavation, completely excavate the structure and take all the materials into a museum. And they did an amazing job, and that's why we this excavation was, I think, in the 80s. So we 40 years ago have been able to do this study because they make a really excellent catalog of all the findings. And Alan Saville was the one who who excavated the site. And as I said, this is a funerary structure. There are 2 funerary chambers at both sides, south chamber and north chamber. Here you can see a picture of the chamber when it was excavated. It's like full of bones. And the other one would be the same. And here you can see a picture of how it might look like while it was in use. Here will be, the door to one of the chambers. This will be this one. So you can see it's huge. This must have been owned by a very rich family, because the archaeologists have estimated that it will take thousands of hours to build this thing. So it must be a family that is able to recruit a lot of people to work with them. So it will be like the funerary structure of a rich family of the time. What do we want to know? So we want to know whether the right to be buried in this tomb was through biological kinship. It was a biological family who was buried here, or maybe it was something more political. Maybe they used this structure to bury all the remarkable people from many different families. We actually have no idea. And if it's actually a biological family, can we study all the social organization of this family? So these were our scientific questions. So what we did, we took 75 samples. They were mostly tooth, like the ones that you are seeing now, and also other type of bones. And we end up recovering 35 genomes. And why is that? Why is much lower number? So some of them don't work. Some of them we try, and then the DNA preservation is really bad, and we are not able to sequence them. And in other cases, it's because several tooth, when we sequence them, we can see that they are genetically identical, because they come from the same person. So that's we pull them together, and then we have more data. So in the end, we were able to recover 35 individual genomes. So these were definitely 35 different people. And what do we do? So very simple. We compare the genome of each person with all the others, and we establish how much of the genome they share. For instance, if these 2 individuals share 50% of the genome, that means they are first-degree relatives. So each of you will share 50% of the genome with your parents and with your brothers, sisters, and with your sons and daughters in the future, probably. And in this case, for instance, they share 25% of the genome. So that means that these 2 are either grandparent with the grandchild, or uncle or aunts with the nephew or niece, and so on. So depending on the amount of the genome that they share, we can establish what was their familial relationship. And this is what we did for the Hasselton individuals. Here you can see all the individuals, and for instance, in this case, this individual that we call SC3M is black. Here is first-degree relative, so it's first-degree relative of this guy who is called NC1M. So you can see there are a lot of very close familial relationship in this tomb. Some of them are not related to anybody. All these individuals here, these are 8, do not appear to be at least biologically related to each other. So why were they buried in this tomb? So it could be like today, we have many people that we consider like our family, but we are not biologically related to them. So maybe they were buried in the tomb because they have a very strong relationship with others in the tomb, but just not through biological ties. But all these others, 27, these definitely were close biological relatives. And what we wanted to do here is to put them all in the family tree, reconstruct the family tree of this family. And we did it with all these degrees of relationship. I did this, I remember it was super fun, because it's like reconstructing a puzzle. So if this individual is first degree of this one, and these 2 are second degree relatives, so you start to put them all in a puzzle, and you go and reconstruct everything. In this case, I was lucky that I only had one possible solution of the tree, because we have so many close relationships here, so there was just one solution. In other cases, you can reach slightly different solutions of the tree, but in this case, I was lucky to get just one tree. And I also use not only this matrix, but also the genetic sex that we get from our data, the age. So the anthropologist, by looking at the bones, can tell you, okay, this is a child, or this is a mature adult, or whatever. They can tell you more or less the age. And also in some cases, we do know the chronology. So we know that this individual was buried first and this other was second, because it was, maybe below the other one. So you can more or less, not always, but you can pretty much know the order of burial. So with all this, I reconstructed the family tree. And here I introduce you to the Hasseltons. And this family is important because it's the first family that we have been able to recover from prehistory. And it's the first time that we are able to look with high resolution at different social patterns of a family in prehistory. And I'm just going to walk you guys through it. So we have the men are represented in squares, the circles are the females, and also the children are the same but smaller. And we have all these individuals with colors are the ones that we have recovered, that we have their genomes. And all these ones in black are individuals that we know they existed, but we didn't recover. So for instance, if we have this guy and we have his father, we definitely know that there was a mother, but we didn't recover her genome. So that's why we represented her in black. And I don't know if you have already noticed, but this family is structured around one man in generation one, that we call it the patriarch, and 4 women in generation one, whom he reproduced, and the descendants of these 4 reproductive unions across 5 generations. And now we are going to be extracting all the different patterns that we see in this family. The first one is quite clear. This society was super patrilineal. Why? Because when we try to connect the individuals in the later generations to the first generation, the connection is always through males. For instance, if we take this individual, how does he connect with the first generation? Is through 1, 2, and 3 males. This other, for instance, through 1 and 2 males. So it's always through males. The glue between the different generations are the men in the family. So the main reason to be buried in this tomb is that you could trace a connection with the first generation through a chain of men. And as you see, this is quite clear. We have 0 matrilineal transmissions and 15 patrilineal transmissions. And also now we are going to be looking at these 3 individuals here with the arrows. And I don't know, you can tell me who are they. So who are the fathers of these individuals? Come on, you guys. Who are the fathers of these individuals with the arrows? Are they part of the family? Have we recovered them? No los hemos recuperado, ¿no? Son estos 3 individuos aquí. The fathers, we don't know who they are. And who are the mothers? Are part of the family? The mothers. Yes, right? Who are they? So they're women who reproduce with either the patriarch or one of his sons. So for instance, this woman reproduced with the patriarch and has 1 son, and she had another son from a different man that we don't know about. So what is the hypothesis for these individuals? Is that the men in the family, the patriarch or his sons, they adopted into the family the sons that their wives had with a different men. And this could be a way to grow the clan very quickly, because not only your biological sons are your sons, but also your stepsons. Tus hijastros también son parte de la familia, igual que tus hijos biológicos. So this is, of course, one hypothesis, but there are other options. I don't know if any of you guys have thought about a different hypothesis for these individuals. Anybody? So it could be an infidelity, right? It could be that the men in the patriline actually don't know that these are not their biological sons. They think that these are the biological sons, and that's why they give them the same right to be buried in the family tomb as their biological sons. It could be unos cuernos prehistóricos. In this case, not by the men, like in these modern cases, but by the women. But of course, we will never know which of the two options, because we can know a lot about this family, but we will never know what was in the head of the men of this family, whether they knew that these were not their biological sons and they give them the right to be buried there anyway, or they didn't know, and that's why they thought that they were the biological sons like the others. We will never know that. So. And the other pattern, very interesting one, is that there are multiple reproductive partners. So we have the patriarch reproduces with 4 different women. One of his sons here reproduces with 2 women, and the 4 adult women that we have reproduce with 2 different men, each of them. But of course, we don't know whether this is polygamy. They were having these reproductive partners all at the same time, or is serial monogamy. Maybe they were, they established a relationship with someone. This relationship ended, maybe because one of the 2 died, or maybe it was like some kind of prehistoric divorce or whatever, and then they established a new reproductive partner. We can know that, but we have some hints that it could be serial monogamy. And the reason why we think that is because we think that, for instance, he had children with this woman much earlier than he was having children with this woman. Why? These are only hints. We don't know for sure, but if we look here, there is this woman who had children with the son of the patriarch from one of the women, and with the grandson of the patriarch with this woman. So that means that probably, she was having children with these 2 men. Probably these 2 men were of the same age, even though they were in different generations. So that could be explained if, the grandsons of this woman are of the same age as the sons from this woman, because he was having sons with this woman much earlier. And we can think about some modern examples of this, where you have individuals that are of the same age, but from many very different generations. And one example is Mick Jagger. You should look at this tree because it's super funny. But this guy has been having children over almost 50 years with 5 different women, and at least in paper, officially, it's been serial monogamy. I mean, I don't believe that he's been completely monogamous his whole life, but at least in the paper, it's been serial monogamy because he's been, there's some period between each of the women. And what happens here is a little bit the same as we have seen. We have here this guy, who is his last son, is basically the same age as one of his grandsons and one of his great-grandsons. So basically, he could go to the kindergarten and pick his son, his grandson, and his great-grandson in the same kindergarten almost at the same time, And this happens when you have kids over a long period of time with different women that even though these 3 babies, even though, well, now they are not babies, but they were a few years ago, even though they are from many different generations, they have the same age. So this is a hint that what we could be seeing in Hazelton could be serial monogamy instead of polygamy, but it's not a super strong evidence. And then, this is just only 2 more. Now we are going to be looking at the woman. Who are the women that were buried in the tomb? We have, the adult woman are these 4, these 1, 2, 3, and 4, and this woman didn't have any ancestor in the tomb. They were women that were coming from other communities and were marrying the men in the family. And then we have this woman here and this woman here, and this one did have ancestors in the tomb. They were daughters of the clan, but they died when they were children. So there's basically a whole group of the family that is completely missing. I know you guys realize who these are. You. Who is missing here? There is a whole group that is missing, that should be there, but it's not. So these are the adult daughters, right? So all these reproductive unions, they have sons who reach adulthood, so they must have had daughters as well that reach adulthood, right? Because you have like the same probability of having sons and daughters, but none of them is buried in the tomb. There are like 14 adult sons of the clan and 0 adult daughters of the clan. So what's going on here? It's what we call, or at least what we think is going on, is a female exogamy. And this means that if the daughters of the clan, when they die, when they were little, when they were kids, they will be buried in the tomb with the parents and with the brothers. But as soon as they reach reproductive age, and we think it's about like 12 years old, they will have to leave the family and go to other communities. And this was, at least in this family, was completely strict, because there is not a single adult daughter buried here. So what we think is going on is that they are leaving the family, and we will probably find them when we analyze other tombs that are like within kilometers of this one. Probably they will be buried there with their husbands and with their sons. And as I say, again, this is, at least for this family, it was completely strict. So you can imagine yourself, you guys are like probably like 15 or 16. If 4 years ago, all you guys have to leave the family, and you cannot decide that. So it has to be done, at least in this family, And then finally, this is the last one, I promise, also very interesting. As I told you before, there are like 2 different chambers, the south chamber and the north chamber in the tomb, right? So we can now paint the individuals here based on where they were buried. So green would be the south, and this yellow-orange would be the north. And what do we see here? So we see the 2 families of the 2 females on the right. In this case, this one we haven't recovered, but this one in the first generation, yes. And all the descendants, they were all buried in the south. And the patriarch, the 2 women in the first generation on the left, and most of the descendants were buried in the north. And what does this mean? This means that even many generations later, this woman in the first generation were remembered, because they were buried in one side or the other, depending on from which of the women in generation 1 they descend from. So this means that even though this society was very patrilineal, the women have to leave the family when they were about 12 years old. Some of them at least were remembered in the memory of this family, even 5 generations later. So at least some of the women were definitely important for the community. And we also know that, this architecture of the tomb, the fact that it was symmetrical, it was not just a pure architectural desire or aesthetics. It's because probably when they were building the tomb, they knew that there was some kind of separation. 2 women form one sublineage and 2 women for the other sublineage, and they will build 2 opposing chambers in the tomb to bury each of those. And there are other tombs like this one where there are like 3 chambers, others have 1 chamber. So maybe in those cases also, this architecture is also related to the social organization of the family. And I think that was pretty much all. I think this is a very beautiful example of the level of detail that we can get with these techniques about social organization in the past. I think it's like, it's like bringing these families back to life again, and probably, they would have never imagined that 5,000 years later we will be talking about all their affairs. But now we can do it with this archaeogenetic techniques. So that was all I had to tell you guys. And if you have any questions, I will be very happy to answer. Or if you have any question in the future, you can drop me an email. I will be happy to answer. So thank you.
No importa. When adult women move out of the clan, so adult men, how they can reproduce. they move to another clan to get adult women?
No, because we say that the daughters are missing, the women leave the clan, but there are other women from other communities that come into the place. So at the end, it's the same. We have these 3 women in first generation and this one that are clearly coming from other places. They are not from the family. So it's like, yeah, exactly. Basically in this society are the men who stay, and then the women are the ones who move. So that in this way they avoid, reproducing with family members because they are exchanging.
They can
the
avoid
women.
genetic diseases
Exactly.
in some way.
Yeah. At least in this case, there was no inbreeding because, as I said, they are like some of the women are coming into this family.
Do you think that the 15 years you have been studying have been worth it, or would you have liked to change all those years in different places and maybe make them shorter than 15 years?
Well, I think in my case, they have been worth it. You are referring most about working in abroad or in other different places, right?
Yes, and also about the long time you have been studying.
Yeah, in my case, it's been worth because I like what I do. When you enjoy what you are doing or studying, and then also doing research on is much easier. So I think they have definitely been worth it, at least for me, because I'm doing something that I enjoy. I have some friends that, for instance, during the PhD, they didn't choose a topic that they really liked, and then you are always going to have problems. It's not going to be like a funny ride all the time. You are going to have problems. So if you choose something that you like, then it's much easier because then you can overcome those problems. But I have friends that, for instance, in the PhD, it was very hard because they actually didn't care nothing about what they were researching. So in that case, it was much harder. But for me, I think it's been worth. I mean, I have had a lot of fun. I have been in many different places. When I thought I was ready to come back and I really wanted to come back, then I was able to do that. So in my case, I've been quite lucky in that regard, I think. Yeah, but that's a good question because it's always as I say, you always gonna have problems at some point. So, but then you have to overcome them. Yeah.
To do a lot of training in bioinformatics, is it right?
Yes.
So that part, perhaps it is the more difficult. At least it seems to me that.
Yeah. So about bioinformatics, so in my case, when I was studying the degree, these techniques were only about to appear. So I didn't get any training on bioinformatics or in programming during the university. I guess that now, at least in biology, they will give some courses about programming, but I don't know. But in my case, it was not that hard because when I got all this data, I really needed to analyze this data, so I had to learn how to do it. So it was much easier because I was learning for some purpose. I was not just taking, a programming course, and they are showing you some things but you don't know how to apply them. In my case, I was just applying them as I was learning. So it's been fun, actually. So it's not super hard because, as I said, you don't have to be a hacker. It's just, you have some notion of programming and of programming languages, and then you can learn from that. So it's actually not that hard, and it's fun as well knowing how to program. So, and as I said, in biology, it's gonna be needed in basically every field, I think now, because now we are moving to having a huge amount of data, a lot of statistics involved. So. Doing manual analysis is not going to last for a long time. So I think in biology, at least, in genetics for sure, it's really, really needed. Yeah.
And now you are working in which project?
Well, now
working in that?
Yeah, I have many projects. I have different projects working on the same social organization, but doing the same in other sites. Because this is just one family, but we don't know whether the patterns that we see here were widespread. Maybe they were only happening in the rich people of the time, but that the common people were not practicing this. So we need to do much more examples to understand what was common, what was just on this family, and so on. So I have similar projects like this in other periods, in other places of the world, and I also have other projects talking more about migration, so understanding in one place all the migrations that have been going on over the years. So, for instance, in the Basque Country, to explain all the history of the Basque Country with DNA. So, yeah, I have several projects about this.